Now, another thing that's important to understand about our base pairs or our bases within a DNA molecule is the instance in which mutagenesis takes place. So when a nucleotide is converted from one base to a different base, this is commonly takes the form of deamination. Now this is a fairly slow reaction, but what can result is C is converted to U. 5-methylcysteine is converted to a thymine, adenine to a hypoxyxanthine, guanine to a xanthine. And so whenever you have these base pair changes take place through this deamination event, well, what's going to happen? What's going to happen is that the base that was there is going to hydrogen bond with a different base. So if you think about this happening in a molecule of DNA, where there was a C, now there is a U. Okay, so that U can serve as a template, but what's that U going to attract? That U is going to be complemented with an adenine. And so where there was a CG is now an AU. And in future generations, that A is going to code for a T and so on. And so you have these mutations that take place where, over generations, you have a difference in base pair and therefore a difference in codon, therefore a difference in the protein that's ultimately coded for. So in addition to that, you can also have this depurination. And so what depurination is whenever you have a guanosine residue in a DNA molecule where, what's that word, depur, okay, we're talking about a purine. A purine is going to be lost through an N-glycosidic linkage being hydrolyzed. So an apurinic residue is going to result from that, and ultimately that DNA molecule is going to have to compensate and basically have a minor deletion there. So other ways in which mutagenesis can take place: oxidative damage. So a guanine can be hydroxylated. Mitochondrial DNA is very susceptible to any sort of oxidative damage. Guanines can be methylated, which when a guanine is methylated, then it's not going to complement for a cytosine. Now, all of these things are things that cells have means of compensating for and basically correcting, but it's not a 100% solution, or you don't have absolute correction of these sorts of mutations. Now, what we have to worry about, and what is very important to consider, is in our environment in which we live, well, we in Texas have high incidence of UV light. And what that UV light does is it induces dimer formation of pyrimidines. Now, this is ultimately a major factor in basically skin cancers like melanoma developing, but it's the result of these dimers forming, and therefore the proper base pair is not going to be introduced in replication or in transcription, so you can run into a problem there. In addition to that, you have ionizing radiation, which causes rings to open and double-stranded pieces of DNA to be fixed. This is actually quite a difficult task to fix. So ionizing radiation from X-rays and gamma rays. I think it was the Incredible Hulk had gamma rays, but I don't think that this ever really results in Incredible Hulk likeness. Now, cells can repair these, but it's accumulation of these mutations that is ultimately going to lead to cancer formation and development. So wear sunscreen. Wear sunscreen. So you need to wear sunscreen. Okay, Now, other functions of nucleotides, this is one that we've kind of overlooked almost. We've talked about it, but haven't gotten into it exactly. So this is really just some terms that you need to be familiar with. We already talked about the lowercase d indicating deoxyribonucleoside 5' phosphates, DAMP, ADP, DATP. We're talking about a deoxyribose there. Our non-deoxy or our simple ribonucleotides would get the AMP, ADP, ATP. So when someone talks about ATP, they are specifying the sugar is ribose. When somebody talks about DATP, they're specifying that the sugar is deoxyribose. Now for the structure of our molecule, well, what this shows is a nucleotide triphosphate, or an NTP, a nucleotide diphosphate, or an NDP, and a nucleotide monophosphate, or an NMP. Those phosphates have different terms based on their proximity to that 5' carbon. Alpha is the first phosphate, beta is the second phosphate, gamma is the third phosphate that is on an ATP or an NTP molecule. Like we previously mentioned, many nucleotides can serve as coenzymes, and this is actually this guy right here. Coenzyme A is one that we actually looked at in the chapter 1 material because of the fact that we were looking at all of the different functional groups. This coenzyme A has a number of different functional groups, and a number of different parts of it are kind of unique in the sense that we've got a phosphate group, we've got we have tons of different groups on it. These other molecule, and sorry, my point was it's got this nucleotide and then this huge modification to it. Likewise, nicotinamide adenine dinucleotide and flavin adenine dinucleotide, they have this foundation of a nucleotide. And then there's these huge modifications on the sides of them. And actually, this NAD plus has a nicotinamide adenine dinucleotide. So here is our second nucleotide, but the second nucleotide, or our nitrogenous base, is not like adenine or anything like that. Our second nucleotide is nicotinamide. So that's why we've got nicotinamide adenine dinucleotide. So there are two nucleotides, and one of them is nicotinamide, and one of them is adenine. So those are all factors in what different nucleotides do. So they also play regulatory roles. If you go on to take a metabolism class, you'll talk about cyclic AMP, cyclic GMP, and PPGPP. You'll talk about these in their regulatory sense of basically stimulating a feedback loop or stimulating a metabolic pathway, either activating or inactivating it. All right, well, that wraps up chapter number, I believe this is 8. I'll see you in chapter 9.